Hello, welcome to VitaSite's webinar on Islet cryopreservation. Um, I'm honored to have Erik from the University of Minnesota give the presentation for a recently published paper that I provided the link in the announcement. As you can see from the announcement, Erik has broad training as a chemical engineer in islet transplantation, transplant surgery, and his research interests at the University of Minnesota are in transplant tolerance and organ tissue cryopreservation. I believe that the work that Erik and his colleagues at the University of Minnesota will be transformational for not only transplant as emphasized in his recent paper, but also in diabetes research. The reason I say that is we've been involved in developing enzymes for human hepatocyte isolation, and I can tell you that in drug development now, all of the hepatocytes that are used in key studies are all cryopreserved hepatocytes. And what it provides these drug developers is consistency, convenience, and using these same cells long term. So I think it's going to be interesting to get your comments on what you see again, a breakthrough in this field will be. And a few ground rules before we start. There will be a poll, one at the beginning, if you can answer the questions or participate in the poll, and two at the end. All the participants are muted. You can ask questions via chat, and you can pose those questions so everyone will see it, or you can go through the administrators. We'll go through your questions at the end of the presentation. And I think that's it. I'll hand it off to Erik and enjoy the presentation.
Thank you so much for that kind introduction. I'll pull my slides. This is really sort of an honor to come and speak to you all, because it's been now a couple decades since I was really involved in islet research back at UCSF, and many of the people in this arena and on this webinar are people that I followed for decades, the research that you've done and the advances you've made. So it's quite exciting for me to be able to talk to you a little bit about our developments. And before I even do that, I want to tell you that this is really a collaborative project between myself and John Bishop of the Department of Mechanical Engineering here at the University of Minnesota. And the two of us have had now about 7 years of working together. Someone sort of introduced us and said that these people over in engineering want to cryopreserve limbs, basically for military people that have injuries. And they said, you do small animal transplant models, why don't you go talk to the people? And I said, well, why don't, doing a leg is going to be a lot more difficult than doing a kidney. And that sort of began a series of projects. And it's really been a rewarding experience to combine some of the engineering that I did in the past and has now come back into these things. And then also islet transplant, which is now coming back into the work that we're doing. So it truly is a collaborative work between myself and John Bishop from Mechanical Engineering, and for our islet project between us and Quinn Peterson, who's a stem cell islet person at Mayo Clinic. So this all began, as you all know, with the Edmonton Protocol. And, there were a couple of things that stood out about this, both the success and also the challenges. As we know, multiple transplants were often required over, a period of time, and that required different surgical procedures and potentially different induction immunosuppression rounds. And so one of the main challenges facing islet transplantation, there are several, but one is the sort of lack of sufficient numbers of islets when and where they're needed. If we could have as much as we wanted at the time of the transplant, we could do single high dose transplants rather than multiple dose transplants. And so The problems, as you know, multiple islet transplants are needed for insulin dependence in some cases. If you need more than 10,000 islet equivalents per kilogram, and you have a 70 kilogram person, that's 700,000 islets. And if we're only isolating 300 or 400,000 islets, that's typically two or three islet donor doses to get insulin independence. We also know that the transplants are not as durable as we would like for a combination of reasons. And one of those might be that if you start out with a marginal number of islets, you have less reserve for injury over time. Multiple transplants mean more immunosuppression, multiple risks. There are two solutions along this line of argument. One would be to pool donors for high-dose transplants, or for an alternative islet source such as stem cell-derived islets, which some of you are involved with. And stem cell islets have similar concerns or considerations in terms of cryopreservation. In our studies, we use human embryonic stem cell-derived islets that Quinn Peterson at Mayo works on developing, and he uses a protocol that he worked on with when he spent some time as a postdoc with Dr. Melton in Harvard, and we use these that as a sort of a model system. It allows us to get large numbers of islets or islet-like clusters at one time for quality control testing, but also is important for this field because one of the challenges that you all may know better than I do is that islet stem cell-derived islet batches are not all the same. Occasionally you get a good batch and occasionally you get a bad batch. Some of them are easy to detect based on the number of insulin-positive cells, but other things, functional assays, can take days or weeks to determine. And one of the problems, just like in native islets, stem cell-derived islets deteriorate in culture. So if it takes weeks to determine if this batch is good, it may have deteriorated to the point when it's not useful at that point. So if we could cryopreserve stem cell-derived islets, we could make a giant batch, test it out, find the good batch, make it a single patient aliquots, and then use that batch for a more economical and efficient way of delivery of those to patients. So our objective was to develop a method for long-term cryopreservation or preservation in general, and there are a number of potential benefits. It could increase utilization. If we didn't have to rely on the ideal donor, and if we were aiming, if we could use all donors, essentially, even if we were getting 150 or 200,000 islets, we could pool those and use them effectively. We could do better donor recipient matching if we bank things. We could match HLA types, things like that, that might have better efficacy down the road. We could do pooled donor high dose transplants. We'd have more time for quality control, and we could make single patient units from large batches of stem cell derived islets. As well, we could use these for tolerance protocols where there's a period of conditioning that may last days to weeks, where you'd ideally like to keep those islets stable while that patient gets their tolerance induction, say through mixed chimerism, and then do the transplant at some time afterwards. So tissue preservation, this comes a little bit from our work in organ preservation. As we know, we can work at all different temperatures. Typically, when we're talking about storage of organs, we're talking about storage, around 0 degrees centigrade, on ice. In organs, we work at a range of different temperatures, or people studying different temperatures. There's normothermic perfusion, where people are pumping oxygenated blood or blood substitutes at 37 degrees, and that may extend preservation time from a few hours to a few days. And, this is sort of what we do with islets, in that we culture them for a period of time. Some people can culture them for weeks, but we know that they also deteriorate over time or change over time. And so we can't culture them indefinitely. Typically, we might culture them for a few days before transplant. We can look at other areas, such as below zero in this area where Arctic animals survive. People do subnormothermic perfusion or supercooled storage of organs, so below the freezing point of water. But really what we want to do is we want to get down here into the cryogenic region. And at liquid nitrogen temperatures, minus 196 centigrade, storage of biological specimens is theoretically indefinite. And so that's really our target for our research. Now we can learn from nature about how nature is adapted to cold environments, and learn some things that might help us in that goal. We can start by looking at Arctic fish. Some polar fish live at temperatures below the freezing point of their blood, and theoretically they should freeze while they're swimming around in the water. the way they've adapted is they produce antifreeze proteins, and those bind to ice crystals and prevent enlargement. So it may have small little specks of ice, but the ice doesn't grow into large injurious forms. So that's how polar fish do it, but they can only go down to a few degrees below zero. Water, I guess, in the saline, so it's a little bit lower than that. That's one way to do it, so prevent expansion of ice. The wood frog, this is the sort of more prototypic thing. The Rana sylvatica, the wood frog, living in northern climates, can tolerate sort of 2/3 or 60% of its body freezing. It goes through a number of metabolic changes, increasing or absorbing urea instead of peeing it out, changing the composition of its cell membrane. So it tolerates up to 60% of its body being frozen, and then upon rewarming restores normal function. Snapping turtle, something we see here in Minnesota a lot. These can undergo or withstand long periods of anoxia. Basically, they take toxic metabolites of anoxic respiration, and they deposit them in the shell, and they basically can tolerate long periods of anoxia. So this is tolerating periods of hypoxia, if you will. And the potentially most extreme are the tardigrade or the water bear. These undergo extreme dehydration. So if they don't have water in their body, there's nothing for them to freeze, and they can withstand real extremes of temperature. So cryopreservation, or storage at low temperatures, and this is sort of along the lines of Dr. Rajad and colleagues. How things began. This began in the 1940s with the idea that if you added a cryoprotectant agent, like we do in the laboratory all the time, we add DMSO to culture media. We can freeze cell lines in culture. You can freeze them, cool them actually to the frozen state, and then thaw them back, and they'll still be viable and functional, at least some of them. However, what happens during conventional slow cryopreservation is that as the temperature drops, you start to form ice outside of the cells. Those chunks of ice start to grow, and whatever solutes are present in the extracellular media are excluded from the ice crystal. So the ice crystal growth formed predominantly of water. The concentration of solutes goes up because those remain in the unfrozen channels around the cells. And because the cell sees a hypertonic solution, water leaves the cell shrinks, and then the concentration of the solutes inside the cell increases. And as we know, as the solute concentration increases, the freezing point decreases. And so they go through a period of osmotic dehydration, and they shrink. The solutes increase in concentration. Eventually, you can go down to very low temperatures without forming ice inside the cell. There's, of course, a balance about how fast you cool them. If you cool slowly, you'll get this cell shrinking and you won't get ice damage, but you get injury from all the solutes around the cells. We call those solution effects. So that's slow cooling. If it goes too slow, you get toxicity from the surrounding solution. If you go too fast, then you can't get this equilibration across the cell membrane. The cells don't shrink, the concentration doesn't go up fast enough, and you get injury from ice. And so we get a sort of an optimal, that's typically 1 degree centigrade per minute cooling that we see in the laboratory for conventional cryopreservation. And Dr. Rajad and colleagues, and many of you probably have tested out conventional methods for cryopreservation of islets over many decades. But it doesn't work as well as we'd like, and there are a number of different reasons. First, we get injury from ice. Extracellular ice injures the membranes. Intracellular ice affects the organelles of the islets, or the islet cells, the beta cells, and then you get injury to the membranes from changes in water concentration. There are a lot of different mechanisms of injury that occur during this process. We get problems or injury from osmotic intolerance during the shrinking and swelling of the cells during washout of the cryoprotectants. That can cause injury. We can get direct toxicity from the cryoprotectant agents, DMSO or other things. We can get chilling injury. That's injury that occurs just by cooling a substance down to sort of moderately cool temperatures. So say going from 0 degrees centigrade down to minus 20 degrees centigrade and then back, that causes injury itself. We call that chilling injury. And then, of course, we form intracellular ice, and that causes damage to the cells. So these are some of the limitations. We still get ice. We get macroscopic destruction of tissue architecture. Well, this may not be critical for islets like they may be for organs. We do get disruption of the islets with ice and cracking and things like that. It does work well for isolated cells in suspension and small embryos. but it doesn't work for longer and more complex biological specimens. And it doesn't work for all cell types because some cells don't tolerate the cryoprotectant, particularly if it's not added and washed out in the appropriate way. So an alternative to conventional slow cryopreservation is vitrification, and that's basically storing in an ice-free or glassy state. And the way that happens is if we look at the sort of the temperature realm that we can go through, we start out in the liquid state. And this would be media that contains biological substances. If we talk about water, we know that the melting point of water is around 0 degrees. If we start above the melting point as liquid and we start cooling, we get below 0 degrees, but we don't form ice immediately. We know ice will eventually form, but it's energetically unfavorable. There's an activation energy to forming ice. And so there's a period or a range of temperature just below the freezing point where water will stay for liquid for very long periods of time, unless something happens, something to perturb the system. Like if you put a bottle of water in the freezer, cool it down to -5, you take it out, it looks frozen, but if you bump it, it will form ice rather quickly. Or if you add little nucleating things like sand or dust or things, it'll form ice. As we cool faster, or we cool lower, we go below the supercooled area into what is written here as the no man's land. That's the space where the activation energy for ice formation is much lower, and the system really wants to form ice, and it will form ice quickly, unless you cool even faster than that. If you cool incredibly fast from the liquid phase, past the supercooled state, through this no man's land, and down to very low temperatures, what happens is the viscosity of the liquid, water or whatever physiologic solution you have, will increase, and it increases to the point when the molecular reorganization that needs to occur for ice crystal formation can't occur. The viscosity is just too high for water to rearrange and form ice crystals, and there you get to what we call the glassy region or the vitrified state, where the substance looks like a glass window. It's technically a liquid, although it behaves like a solid, and you don't get the injurious formation of crystalline ice. And so that's the vitrification process. This was demonstrated in the 1980s by a cryobiologist by the name of Greg Fahey, where he was able to vitrify rabbit kidneys in this case, and this sort of led to a whole sort of many decades now study to try and vitrify different specimens and recover them. Now the problem with that is you have to cool incredibly fast, and further you have to rewarm even faster than that. The rate of rewarming is about 10 times greater than the rate of cooling, and the way that we can achieve that or make it practical is to add various things that inhibit ice. We call these cryoprotectant agents, and really the best ones are mixtures of these. So there are a number of different cryoprotectant agents. There are ones that penetrate the cell membrane, such as DMSO and ethylene glycol. These are really good ones. And there are ones that don't penetrate the cell membrane, sucrose and trehalose, polyvinyl peroxidone. These are extracellular cryoprotectants, and often it's a mixture of these that are used, partly because it's thought that the permeating ones like DMSO are likely more toxic to the cell itself. And so we prevent ice outside the cell, but we also have to prevent ice inside the cell. And to tell you how effective these compounds are, we can look at various compounds. So 6-molar glycerol was one of the original cryoprotectants found in the 1940s by Pulginal, looking at chicken sperm cryopreservation. That has a critical cooling rate of 85 degrees centigrade per minute and a critical warming rate of 32,000 degrees centigrade per minute. So you have to rewarm very quickly in order to avoid ice while you're rewarming, because you can form ice still while you're rewarming because you're in that zone of danger. These are 3 different generations, DP6, VS55, and M22, with increasing concentration of CPA cryoprotectants. And this most recent one, M22, developed by Greg Fahey, is really quite good as a vitrifying agent. You only have to cool at 0.1 degrees centigrade per minute and rewarm at 0.4 degrees centigrade per minute. The problem is it's very highly concentrated, 9.3 molar, and that means it has a risk of toxicity. And so for all of these things, what we're looking at is how fast can we cool and how fast can we rewarm, and then how much CPA do we need. And so the faster you're heating and cooling, the less CPA and the less toxic you have. The slower you cool, the more CPA you require. This requires more loading time to get the CPA into the tissue and in the cells at higher concentrations and higher loading temperatures, all with more toxicity. And so this is one of the critical balances that we face for islets. So we tested a number of different ways to vitrify islets, and other people have done this as well in various iterations. We started out with some traditional things. One is using a cryotop, which is basically something developed for reproductive medicine, assisted reproductive medicine, where you have this little device that has a little droplet of cell-containing solution that's immersed in liquid nitrogen, cools relatively quickly, and then when you want to rewarm it, you directly immerse that in media. It can heat around 90,000 degrees centigrade per minute, so pretty warm, but it's a really slow process because manually you have to do each droplet. We first looked at something called what we call pick and print with laser nanowarming, and what this did was this took we had our islets in a dish, and we had an automated system that would scan the dish and have a pipette tip that would dip in and suck up islets, and we would make a suspension of islets, and then we would print those islets on a copper dish floating on liquid nitrogen, and I'll tell you why that was required in a second. And we could form these little BBs of islet-containing BBs. In order to rewarm them, what we did was when we vitrified them in the presence of little gold nanorods, and those gold nanorods absorb energy when you hit it with the laser. And so if we shoot a near-infrared laser at the little BB containing islets, we can rewarm them incredibly fast, millions of degrees centigrade per minute. And this really works well because you can use very little CPA or maybe no CPA at all. And this was the initial thing that we were trying to do. The problem is it's a little bit unyieldy on this thing because you got to move these little BBs onto the system, and we can do it one at a time manually. We were spending time trying to figure out some sort of flow cytometry kind of based thing, but it had to be done at cryogenic temperature, so it was going to be quite challenging. And so we continued to do that, but it had some problems, which I'll talk about here. The first was the printing. If you try to print directly into liquid nitrogen, the liquid nitrogen vaporizes around the droplet, and you get what we call the Leiden frost effect. It's called the Leiden frost effect. That vaporized liquid nitrogen insulates the droplet, so the cooling is too fast to vitrify. And you can see these are the little BBs. If you print directly in liquid nitrogen, you can see they're sort of opaque because they're little crystals of ice in them. Whereas if you print on copper, and actually copper that has a little mesh over the top, Kim wipe or something like that, you don't have that same Leiden frost effect, and you can vitrify little BBs. You can see these little clear things that are about 1 millimeter in size that contain islets. If you have islets here, and so we were doing that with little vitrified islets, and then we shine the light on it, and basically it would rewarm quickly. This plays fine in the back there. It's a little slow to start. So this system was really developed for other tissues, for Drosophila and embryos and the like. And what happens is there's a timed pulse of laser light that goes down. Yeah, I think it's playing. It takes a little while to get into it. It doesn't want to play, but basically what happens is this, in a sort of fraction of a second, the laser hits this, and it really rapidly rewarms things, and the islets recover. There are some problems, though. Depending on the size of the droplet, these are different outer diameters of the droplet. You get a lensing effect where the laser hits here, and there are areas of shadow in this area because the light doesn't refract in that direction. And so you get higher heating up the top, slower heating down here, and even slower heating at the side. And so there are sort of cold spots in it. When we did this with cells, we could get very high viability, 90+%. But it seemed to be a bit unyieldy for islets, and so we sought to do another method. And this other method was developed when we were trying to cryopreserve Drosophila here. Drosophila are hard to cryopreserve. And what we found is that if you take Drosophila embryos and you put them on a mesh, and then you immerse that mesh into liquid nitrogen, you can cool pretty quick, but you can't cool fast enough. But what Lizon, one of the postdocs in the lab, found is that if you then take this Drosophila embryo, put it on the mesh, and then wick away all of the excess CPA, so it's essentially just a small layer of CPA around the Drosophila, then immerse this in liquid nitrogen, the cooling rate is incredibly high. And then the warming rate, when you dunk that now into warm media, is also very high. So you reduce the mass on the dish, and then that increases the cooling and warming rate considerably. And so this is the approach that we use for islets. We use a different size mesh with a pore size about 36 microns, but this is what we did for islets. And this is sort of a brief summary. So in the traditional way that we do it, we would load the islets with cryoprotectant agents in a tube, put them on a mesh, and then dunk that mesh in liquid nitrogen. The problem is all the CPA around it slows down, has thermal mass, and slows down the cooling rate and the subsequent rewarming rate. And so that wasn't fast enough cooling or fast enough rewarming. So we went to this wicking process where we basically took the islets that were on the mesh, we wick away all the excess islets, immerse that in liquid nitrogen, and we can cool very fast, and then store them in liquid nitrogen, take them out, stick them in warm media, and then rewarm even faster. And so that actually works really well. So this is sort of a summary of sort of how things work. We work in a number of different host species. We work in pig, mouse, human, and now non-human primate islets, native islets, as well as embryonic stem cell derived islets. We isolate the islets. We balance the amount of CPA in the loading formation, the loading protocol, if you will, with the amount of ice that we get. And as we cool and heat faster, we can shift that balance to use less or more CPA. We store them. We stored them for up to 9 months, but theoretically they can be stored indefinitely in a vitrified state, and then assess them in various forms. And this is basically a schematic showing what happens. We load them in tubes. We put them onto this mesh. We wick away the excess CPA. We immerse them in liquid nitrogen. And then for rewarming, we do the opposite. We put them back into media, and that rewarms fast. The cooling rate is incredibly quick, about 50,000 degrees centigrade per minute, and the rewarming rate is even faster, 300,000 degrees centigrade or faster rewarming. So this is the critical part of why we achieve success where other people may have not achieved as much viability afterwards. Now this is talking about vitrification and rewarming and not conventional slow cryopreservation, but this is a conceptual plot showing how much CPA you need, the weight percentage, versus the critical warming rate that you can achieve. And these are other groups that have tried vitrification and rewarming. If we look at the concentration of CPA, say this group, we look at the concentration of CPA they use, about 45%, and we look at the rewarming rate that they use, a little bit over 100 degrees centigrade per minute. We know that that's in this range where we'll form ice. We need to be up in this range. You can either add more CPA and push yourself this way, or heat faster and put yourself this way. So these people likely formed ice when they rewarmed it, whereas we aim to be here. Faster than the critical warming rate of the CPA, which we used, was about 44% weight percentage. And I'll talk about that briefly in a second. So the cooling and rewarming, this is from our Oregon work, where we know that as you start in a liquid zone and cool into this no man's zone, and then down to the glassy zone, you have to cool faster than this critical cooling rate. Again, this is for kidneys in this case. So you have to cool at a fast rate. And then the warming is the same thing. If the black dotted line is an indication of the warming rate, you have to heat everything warmer than that critical warming rate. So the warming rates that we saw, if we just did convective warming without the cryo mesh, we could heat at about 10,000 degrees centigrade per minute, or cool 10,000 degrees centigrade, and rewarm at the same, around 12,000. When we do the cryo mesh that we talked about, we can cool at 53,000 degrees centigrade per minute and rewarm at 300,000, and ultimately we get high viability, 92%. And I'll tell you about that in a second. So one of the critical things is how we load the CPA agents, and you can't just add them just directly. You have to do it in a stepwise fashion because they're hypertonic. So we developed a system for modeling the diffusivity using a small little flow chamber, and we could then develop a protocol for stepwise loading and unloading of the CPA that incorporates toxicity cost function, basically how much toxicity you get from the CPA from exposure times at different temperatures, as well as the volume change that occurs when you expose them to hypertonic or hypotonic solutions. So there's a range of volume considerations that you use. And with this, we developed a sort of an optimized protocol, which could achieve the concentration that we wanted for each step, but minimize the toxicity. And that's how we achieve success. So Our biological assessment is we use acridine origin perpetuum iodide. It works well for us. We either do confocal microscopy, or we dissociate the islets after treatment and then use a fluorescent cell counter to count the percent live and dead. This is quite a busy slide, but I'm going to simplify it. Basically, I want you to focus over here on the right. This is comparing different CPA agents that we looked at. When we started with propylene glycol, you can see this is the viability of just loading and unloading the CPA alone. And then the next bar, the green bar, is what happens after you vitrify and rewarm with that CPA. You can see that propylene glycol has too much toxicity. The viability is down to 25% just with CPA. When we switch the other things, ethylene glycol, the viability increases. There's less toxicity. But we see a change when we go from just CPA loading to vitrifying and rewarming. We're seeing a decrement in viability. When we do mixtures, we get less toxicity. A compound of 44% that's 50% PG and 50% DMSO, or 22% by weight of each, is much less toxic than 44% of either alone. But these all sort of had substandard, or what we say, not optimal viability, until we got to this mixture of ethylene glycol and DMSO. And what we found is we could mix the two of them together and get very high viability after loading and unloading with their protocol. But if we use 16% ethylene glycol plus 16% DMSO, we got a decrease in viability when we vitrified and rewarmed them. We presume that we're forming ice, and so this doesn't have enough glass forming ability to prevent destruction from ice. When we stepped up to 22% of each agent, then we got high viability, so little toxicity, and little change when we vitrified and rewarmed it. And so this became our CPA agent of choice. When we went higher than that, we got more toxicity from the CPA. And I'll just point you down here. This is islets. This is stem cell derived islets now, vitrified and rewarmed. This is CPA only and vitrified and rewarmed with that CPA, 22% EG, 22% DMSO. You can see that they're mostly green, so those would be live. Whereas propylene glycol, you can see that they're mostly red, where you get a lot of death in that. So that was our CPA moving forward. This is just quickly showing you that even after vitrification, there are some changes that occur during the process, and they require a little bit of we put them back into culture for a few hours before we test them, because what we see is that this is TMRE staining, which shows mitochondrial membrane potential that increases for over a few hours of culture, so it definitely restores mitochondrial function. The same thing is with ATP. ATP levels are reduced with vitrification, and then after rewarming, if we put them back into culture, the ATP levels recover, and they remain doing that for at least 24 hours. So we typically use 3 hours of post-rewarming culture before we assess them for function. So when we look at them after nanowarming, or after cryomesh rewarming, we can see that morphologically they look quite good. The vitrified and rewarmed islets have a smooth border. Histologically, they appear very similar to the control islets, whereas conventionally cryopreserved islets do have damage and destruction of the architecture. When we look at with electron microscopy, we can see that there's the mitochondria are preserved, the membranes of the nuclear membrane and cell membrane are smooth, and the vesicles, the granules are preserved. Whereas if you do conventional cryopreservation, the membrane of the nucleus is roughened, same with the cell membrane, and there's some swelling of the vacuolization of the vesicles inside the cell, suggesting dysfunction. We look at a number of different species: mouse, human stem cell derived beta cell clusters, pig islets, human islets. They all seem to function well, work well with good viability with vitrification and rewarming, and relatively poor viability with a conventional cryopreservation, at least in our hands. We can store them for at least 9 months, and they seem to maintain their viability similar to what they were at time zero. When we look for apoptotic markers, we look for tunnel stain. These are the upper ones, are tunnel stains and annexin translocation. We can see that there is a little bit in the center of the vitrified and rewarmed islets, a little bit of tunnel positivity, and a little bit of annexin translocation, suggesting there is some DNA fragmentation and apoptosis going on, but it's much less than conventional cryopreservation, where there's much more apoptosis and more central tunnel positivity, suggesting DNA fragmentation. We look at beta cells and non-beta cells, and I'll just have you focus here. On the red are the insulin positive cells, and the right are the insulin negative cells. We're looking at the relative viability. The relative viability of the beta cells and non-beta cells was equal. We look at metabolic recovery afterwards. Again, this is TMRE staining with the various islet types. We can see that, again, that after recovery, we can see that there's intense staining with the TMRE dye, suggesting mitochondria are present and functional, whereas conventional islets have a central lucency, suggesting that they have slower recovery of metabolic function. The same is true for ATP. We see some decrease compared to fresh islets, but it's much less than the conventionally cryopreserved islets. So a mild decrease in ATP with 3 hours of culture after rewarming. We use seahorse assays to look at oxygen consumption rate for various metabolic parameters. We can see that control and vitrified and rewarm islets perform very, very similarly. This is the oxygen consumption rate in response to various mitochondrial stimuli, as compared to conventionally cryopreserved islets. The oxygen consumption rate is much lower, and dead islets are treated with ethanol are much lower. When we pool all the different parameters that we can measure with the seahorse assay, we can look and see that almost all measures of metabolic function or oxygen consumption for metabolic function, such as basal respiration, maximal respiration, respiration for ATP production, they're all fairly equivalent between control islets and vitrified and rewarmed islets, suggesting impact metabolic function. We look at function in vitro. This is GSIS assays. We look at the mouse stem cell derived beta and human islets. We can see that with exposure to increasing concentrations of glucose, or KCl for membrane depolarization, we can increase in insulin secretion. And what we see is that there's very little difference between control islets and vitrified and rewarmed islets in terms of their insulin response to glucose stimulation. So they function in vitro. They also function in vivo. These are islet transplants and syngeneic recipients, so 250 islets per recipient in SDZ, streptozotocin-induced diabetic mice. And you can focus on the lines down here. These are fresh control islets versus vitrified and rewarmed islets. And in 9 out of 10 in this group, we had normalization of blood sugar within 2 days and excellent glycemic control for the 150 days that we tested it. There was one technical failure where the islets leaked, and so it didn't completely recover, but did maintain at least a relatively steady glycemic control. If we take the kidneys out after kidney capsule transplant, the hyperglycemia recurs, showing that this was indeed due to the transplanted islet. So this shows that they function in vivo. We can use 9-month stored islets or recently vitrified and rewarmed islets. They all work the same. We do IP-GTT assays, looking at the insulin response to glucose challenge in vivo. You can see that the fresh control and vitrified and rewarmed islets function similarly, versus conventionally cryopreserved islets don't have the same glycemic response. Area under the curve was the same. When we look at transplant in non-skid gamma mice, these are now human stem cell derived beta cell clusters that are vitrified and rewarmed. We can see that they produce human insulin. And then when we do a challenge, this is red versus blue, we can look at fasting insulin versus stimulated insulin measured by human insulin. We can see that conventional islets don't have a real big response. We do see some insulin, but it doesn't respond to glycemic control, whereas the vitrified and rewarmed stem cell derived islets do have a stimulated response with increased insulin. When we do confocal microscopy, we can see what we would expect to see. This is insulin staining, glucagon staining. In the syngeneic mouse transplant, we can see strong red in live control and vitrified and rewarmed. A little bit lower amount of glucagon staining, but still preserved glucagon positive or alpha cells. In the xenotransplants in pigs, we see insulin positivity and glucagon. In the stem cell derived beta cells that don't have alpha cells, we only see insulin. When we compare those to conventionally cryopreserved transplants, we see very little insulin and very little glucagon. There is some there, but it's much less than with the vitrification rewarming. The recovery of islets is excellent. Typically 97%, and we're just working now on our scale up. We've done a few assays and initial studies with 10,000 islets. We can now increase this even further, where it was a little bit decreased, but typical recovery is about 97 or 98% when we're working on that sort of the 2,000 islets, 2,500 islets. It's slightly reduced to around 94% with 10,000 islets. And this is probably just a handling thing as our postdoc was getting used to. So future goals. We have biological goals to understand the immune response to vitrification rewarming. We want to look at the transcriptome changes that occur. We're working towards large animal models and preclinical translation. And we want to incorporate some tolerance protocols. We have a concerted effort for scaling up. One of the big benefits of this process is because it's essentially a two-dimensional process. We don't really care where the Z dimension is. We just need to make the mesh bigger to do more islets, or we do stacks of meshes. And so in this case, we sort of do 50,000 islets per mesh, and we can stack them 10 stacks per container, do 500,000 in a batch. And so we think this is how we'll get to a clinical scale. And this is working very well with different types of meshes that may cool and heat even quicker. So in summary, the islets tolerate a CPA of 22% EG and 22% DMSO. We get fast cooling and rewarming. The viability is high, typically 90 to 92%. Recovery is high, 92 to 98%. They preserve morphology and respiration and function in vitro and in vivo, and it works for a number of different species. And we think it's scalable, although we're working on demonstrating that at true clinical scale. So in comparison to other techniques in the past, conventional cryopreservation was initially developed by Rajad and colleagues, in the 70s. It had not great viability and not great throughput. They modified the CPA, and various groups have done the same thing, for improved viability and function, but not really scalable. When they changed the system to cryobabs for increased scalability, that was at the sacrifice for viability and function. A number of groups switched over to vitrification, and the problem is none of them were able to achieve the combination of high viability, function in vitro, function in vivo, high recovery, and clinical scalability until sort of our protocol came along. We incorporate a lot of the developments that other groups did, but we were able to sort of put it all together in something that really worked. Those other groups. This is just a gross advertisement for what we do. This is sort of taken on my life. We work in cryopreserving a number of biological specimens, from tissues and transplants to cells for drug discovery, vertebrate models, a coral species for biodiversity, some aquaculture for foodstuffs, skin, and even potentially whole body things. And this is all sort of housed under the NSF-sponsored Engineering Research Center at our institution, and my colleagues that were involved in this research, which I'll highlight, particularly John Bischoff in mechanical engineering and his postdoc liaison, and my postdoc Joseph Rao. Those are really the main people that did the bulk of this work, as well as our colleagues at Mayo who provided the stem cell-derived islets. And with that, I'll be happy to answer any questions that you have.
Yep, thank you. Thank you very much, Erik, for a superb description of what vitrification really is, and also to show all the work, extensive work you've done to prove how this method is superior to ones in the past. I think we have one question that you may want to address from Hazen.
How can you ensure sterility? This is a really important thing, and this is one of the areas that we're moving into. So there is direct contact between the islets and liquid nitrogen. You can sterilize liquid nitrogen by either producing sterile liquid nitrogen, which is challenging, but you can be done with filters, or you can irradiate it with gamma radiation, or some people in the reproductive biology field use UV light to sterilize it. But the problem with that is that you may be sterile, but you have to then check for endotoxin. It's harder to check for endotoxin in liquid nitrogen. So that's something we're working on. The other thing is preventing cross-contamination between samples in a large liquid nitrogen tank. As we know that there's a challenge in the reproductive biology field, if you will, to sort of maintain things and not have contamination between different tubes. And these are all things that we haven't even addressed yet, but that's what we want to do.
Does it have to be in liquid nitrogen? I mean, can you use these really ultra-low coolers that get down to liquid nitrogen temps?
So the initial cooling is in liquid nitrogen, but
right.
They could be transferred to a -150 freezer for storage.
Right.
We have been able to do it for like I said, 9 months If we want to go longer than that, potentially there could be, desiccation, if you will, or injury at the surface of the islet, but we don't know as much about that.
Okay. Another question I have is, I view this, I know you're on the transplant side, and I know that you've gone through this figure in your figure S12 that was in your paper about how you could scale this up and get a lot of islets. I foresee that you'd almost have islet isolation centers, and the question is how do you ship the islets to those centers? And I would imagine that you could make the rewarming pretty foolproof, because that's really all the user needs to have if they want to start doing this, right?
Yeah, so this is really one benefit of this technology as compared to, say, our organ preservation technology, which requires $500,000 piece of equipment that takes 480 volts in a room to isolate it from potential electromagnetic fields. So this is basically, and once it's vitrified, and even the vitrification process is not entirely complicated itself, but it's taking it out of the container and dipping it in warm media or room temperature or 4C media. The islets are pretty well stuck to the mesh, but then they come off as they rewarm and you wash it out. So, we have to work through all the details, but conceptually you would have a bank of these in freezers or liquid nitrogen tanks, send them in a shipper, and then they would be taken out. There'd be some degree of training because you have to sort of do it efficiently. So you do it
right
into the media at the same time. And that was one of the limitations when we went to from 2,500 to 10,000 islets, is how to get the mesh into the liquid nitrogen, quickly and things like that. But I think with minimal training, you could do it. You could cryopreserve them, at a center of excellence, or whatever you want to call it,
Yes, exactly.
Store them wherever you want, ship them in unit batches, and rewarm them at the transplant center. There's sort of ethical and policy issues related to all of the work that we do, which has been a fun part of considering this about, who becomes a center, what's the cost, what's the extra cost for this compared to the conventional, and what is there a benefit to that extra cost, and what are the regulations which struggle with islet transplant already.
Because I think that I counted one time, there's 22 islet training or research centers in the United States. 22 diabetes training or in research centers. And, I would think this would be invaluable to some of these centers, and you could even use less cells. And you could use, have centers isolating type 2 islets from type 2 diabetic patients, which I know I think it's NPOD does already, which have tremendous ability to start now studying instead of craziness and trying to study as much as you can before the islets die. You could think about the experiments, so to speak.
You had 500,000 islets at once, and you got to use them all in 3 days.
That's exactly right, and that's depending on how many you actually got, right? Which is another.
So there are other minor things related to sort of the islet isolation that many of your participants may be interested in. That is, as it's been a while since I've been involved in this, but it's a balance about the islet recovery versus purity. And so this, if you're willing to sacrifice on recovery a little bit, as you're doing density gradient separation, you could sort of say, okay, I'm gonna only get the pure fractions, sacrifice a little bit of viability, transplant the more pure populations, which we think function better with less inflammatory response, less mass, so less portal hypertension. Now you may guarantee yourself needing a 2-donor transplant or a 3-donor, or as Dr. Zapp did, 10 donors, I think, in one of his papers where they pooled donors back when they were studying it. You could pool donors for efficacy. Now, the balancing thing is that now you're exposing to a broader repertoire of HLA molecules, and so maybe there's allosensitization. But if you're requiring multiple transplants anyhow, you're going to have that you could do a single high-dose transplant with greater immunosuppression, minimize your initial allosensitization, rather than doing subsequent transplants at lower intensity immunosuppression, induction of immunosuppression. So a lot of conceptual ideas about it.
There's a question on the chat. If you switch to the chat button from Dr. Sweet.
Oh, there we go. Is it possible to load the mesh inside a cryobag for downstream applications? What do you think the thawing problem? So that might be one way to. We're really, as I showed, really at the conceptual thing, we're still working on what is the optimal mesh, and then how do we do it to make sure that when we put the islets, and when we do it in a small scale, it's easy for us to spread them around. When you do it at a large scale, they can sort of lump up on top of each other to make it a little bit thicker and slow the cooling and rewarming rate. So we're trying to figure out how to get a good way to get sort of a good monolayer of islets on the mesh. But imagine if you could even put a cryo bag with liquid nitrogen in it, stick them in there, do the vitrification in the cryo bag, seal it in some way, maybe drain the liquid nitrogen off, and then have a way of rapidly introducing warm media. It does have to be pretty quick, but it might be that you could figure out some way to more efficiently do it that keeps a sterile barrier. The rewarming is thawing, essentially. So the other question is, does the rewarming refer to thawing in your presentation? So rewarming is thawing, essentially. But we sort of when we use these terms precisely, since we don't truly freeze them, we often sort of don't call it thawing, and we don't call it heating. We call it rewarming. And so the engineers, they're always after me for being precise with the word terminology. So we're typically calling rewarming as the process of recovering from the cryogenic state to above the melt.
So has anyone, obviously this is, what you're saying is the vitrification is a pretty complicated setup, or?
It's actually not. So, sometimes people don't advertise the simplicity of it. When we were first developing it, the device that we used for manipulating the mesh was just nylon mesh with a 36 micron pore size. And the little handle that we used to reduce the thermal mass was essentially one of those straws we use for weighing chemicals in the laboratory. That sort of, we cut a notch out and super glued the mesh to it. And so we could just dunk it really quickly in liquid nitrogen. So when you go to larger size scale, we're talking about sort of 4 by 6 centimeter now for the scale up size. Then you have to have a bigger container of liquid nitrogen and have a way to get it in there efficiently. But that just requires sort of a practice of getting it in there and a little bit of thought about, is there some sort of support structure around the rim of the mesh that doesn't add thermal mass or as much thermal mass?
I think there's another question.
What was your average culture time before freezing it? And do you see functional difference if it's shorter or longer? That's an excellent question. So when we did mouse islets, we first started with really fresh islets. They got preserved and then we would just use them that day. Then we realized it was more efficient for us to do the experiment the next day. So then we just did 24 hours of overnight culture, and we would do them the next day. Then we started looking at human islets. The stem cell derived islets got shipped from Mayo to us. So we use them. Those could be cultured for a week before they started to deteriorate. So we typically would use them within a week of where we got them. And then we started using purchased human islets from one of the companies that may be participating here, and they send them, and we would use them for a few days, 3 or 4 days before we would do it. So what we know is that if you start out with longer periods of culture, the overall viability decreases slightly in culture. And so the relative viability is maintained, but the gross viability, the absolute viability is decreased because they start out a little bit lower. For almost all islets, except for porcine islets, which were more fragile under our hands, and the longer you cultured them, the less well that they would do with handling after that. Thanks, Craig.
Did you ever think of, have you ever can use encapsulated islets at all? Because, you know, you could, in the old days when people were doing porcine islets, they could keep encapsulated porcine islets in culture for long periods of time.
Yeah, we haven't tested that. We don't really have an active encapsulation program at our institution. And the problem is, as you mentioned, there's not a whole lot of funding these days and some of the sort of more basic islet things. And so unless the way we got non-human primate and porcine islets was that they were offcasts from the
Yes.
you know, other experiments. And so we sort of depend on other people to be doing things to get their sort of excess. And it's kind of slower these days.
And have you transferred his other people transferred the technology at this point in time? It's still working it out. I mean, in other collaborators, so to speak.
Yeah. So right now we're doing it all at our institution, but part of our plan is to show that we can ship them and do them between institutions. In our ERC, we have a group of close collaborators at MGH, and the postdoc who started this has moved there. So now we're going to send them to him. And
oh, very good. Yeah, excellent. So if there's any other questions before we break? If not, I encourage you to fill out the last two questions in the poll. And this was, I mean, phenomenal. I think, again, you're on an exciting path, because I think this, again, to my mind, transformational. If the technology we can start using more consistent islets and do what they do in human hepatocytes with having a much more convenient source of a reproducible set of biological materials you can use for long term. And again, this has tremendous ramifications for also if you had better controls on assessing islet potency for transplant. We could do rapid testing. We had a control material and it'd feel safe. I think it'd be a nice, fill a nice gap where we don't have a real good solution for, say, a potency test for an allo islet transplant.
Yeah.
Or allo islets.
It's linked with the last questions. How far are we in the conversion to a GMP process? And as I mentioned, we're still trying to get the scale up down, and we're just getting good results now. The next steps are sterility, endotoxin, identity. We know that they work really well. We don't know the exact biological equivalence between a fresh and a cryopreserved islet. So we know that 250 islets works in a mouse transplant model, but we don't know if 150 islets. What is is it 1-to-1? Is it 0.7-to-1? What is the decrement? But then we know that, as you mentioned, there's variability even amongst fresh islets, and so if we know that the various bioassays, new mouse bioassay or whatever xenotransplants that we do to test islets take weeks, and so if we could
I think immediately that's just obviously the ultimate step is for you to have frozen donor banks, right, with known haplotypes. But and as an intermediate step, this would be add a lot of value for people who are want to assess potency more rigorously, and then having control material where it's almost like I hate to say a clinical chemistry assay. Your variability with the islets for certain, say, glucose stimulation, and you can normalize those results. So, Erik, thank you again very much for this wonderful webinar.
Thank you. Thanks for the opportunity.
I really appreciate it. Okay, I'll let you go, and thanks again.
See you later.
Yep, bye.
Bye.